So good morning everyone again, and welcome to the NEXTalk webinar titled Decoding Cellular Complexity from Single-Cell Multiomics to AI-Driven Spatial Biology. Thank you all for joining us today. My name is Meng T, and I will be the moderator for today's session. Before we begin, here are a few things to know. We are delighted to have two distinguished speakers with us, Dr. Benson from 10x Genomics and Dr. Mai Chan from A-Star Singapore. We will have two talks in today's NEXTalk session, which will last for about 1 hour and will include Q&A sessions between the talks. If you have any questions during the presentations, please feel free to submit them in the chat box and we will address them accordingly. Please note that this session will be recorded for Dr. Benson's presentation only. As Dr. Mai Chan will be sharing some confidential data during her presentation, her session will not be recorded. We kindly ask the participants to not take any screenshots or capture any images during her talk. Lastly, please ensure that your microphone remains muted throughout the session. Now, it is my pleasure to introduce our first speaker for today. Dr. Benson is a science and technology advisor and 10x Genomics in Singapore, where he covers the SEA region and India. He has a background in molecular genetics and holds a PhD from the University of Melbourne, Australia. Prior to joining 10x, Benson has held various roles in the biotech industry, including FAS manager and senior FAS at Rare. Before transitioning to the industry, Benson dedicated over 10 years to the academic sector, where he published several papers and delivered research presentations at international conferences. Today, Benson is going to present on assess the richness of biological complexity with single-cell and spatial multiomics. Please join me in welcoming Dr. Benson. Benson, the floor is yours now.
Thank you for joining. All right, okay, let's get started. Right, good morning and thank you for joining. Thanks Ming T for the kind introduction. Quickly, my name is Benson. I'm the Science and Technology Advisor at 10x Genomics. And I just want to thank you for joining us today because I think this is the very first webinar, correct me if I'm wrong, Ming T, that we are hosting together with NextGen. So I'm very glad that we have a pretty decent turnout for today's webinar. So for my presentation today, right, I'm actually going to give you a very brief and high-level overview of all the different technologies we have at 10x Genomics. And I will not go through in very technical details of the chemistry and also the workflow. But of course, if you have any questions along the way, please feel free to write them in your in the chat box, and then I will be happy to take them on. All right, let's get started. So this is our forward-looking statement. So if you need to review this, please reach out to us after this webinar. So as we all know that biology itself is actually very, very complex. While we have made very good progress, but we actually are very, very far from finding the final answers due to the immense biological complexity. So therefore, mastering biology requires significantly greater resolutions and massive scale to achieve breakthroughs, like for example, curing cancer, eliminating autoimmune diseases, and also comprehensively understanding the brain. So at 10x, we have currently 4 different toolkits or 4 different technologies that I will introduce today. So the first you see here is our Chromium single-cell technology. So our Chromium single-cell provide all the needs you need to do with single-cell analysis. We have the Vision Spatial. So Vision Spatial allows you to interrogate the whole transcriptome across the tissue in the spatial context. We have another spatial technology which we call the Xenium. So the Xenium is an in situ technology where we actually provide imaging technology to detect where the transcripts are located in your cell within the tissue in the spatial context. And finally, I'll talk about our latest technology from 10x Genomics, which is the Artera. Atera is another in situ technology. So I will actually talk more about this towards the end of my presentation. So let's get started with Chromium single cell. So at 10x, we actually catalyzed the single cell revolution. So we have been around in the field for actually more than 10 years. So we are 10x is actually founded in 2012. So our very first commercial product was launched in 2015. And in 2016, this is where we actually launched the very first high throughput single cell gene expression analysis solution. And since then, we have catalyzed these single cell revolutions, and we actually have the largest and broadest menus of assays and application for doing single cell. On top of these, we also have the best performing data quality for a single cell solution. So if you look at some of the figures you see below here, right? So to date, there have been more than 15 billion cells being profiled using our Chromium technology, and this has led to more than 9,000 high-impact publications. So 10x provides a very comprehensive multi-omics capability. So we have assays not just looking at gene expression, we have assays looking at chromatin accessibility for epigenetic assays, like single-cell A-testing. We have solution for that. We have assays to look at the T-cell, B-cell receptors. So if you're interested in looking at the T-cell, B-cell sequence, right, we have solution to do that. We have assays to look at cell surface proteins, high throughput CRISPR screening, antigen specificities for your antibody studies, and even intracellular proteins are all enabled with our Chromium solution. And on top of this, we also have acquired a company called Scale Bioscience. So Scale Bioscience has an advantage that they have solution to do single cell methylation. So we are actually providing single cell methylation as part of our single cell solution. And with regards to sample types, our assays are basically species agnostic. So not just human or mouse or rats. So if you have any species, right, eukaryotic species, theoretically, these will be compatible with our Chromium solution. And we also have continued to develop a lot of these protocols and guidelines to handle those very difficult to deal with sample types. So not just cell suspension or PBMC, we have now protocols that can deal with clinical samples like whole blood, fixed tissue, FFP tissue, and even organoids is also compatible with our Chromium solution. So how can single cell tools be applied to your research, right? Single cell has the ability to reduce cellular heterogeneity, right? So if you are doing a bulk RNA sequencing approach, right, heterogeneity will not be able to be deciphered with a bulk approach. It's only through single cell analysis that you can actually work out what cell types are presented in your sample. And even within the cell type, right, the different states of this cell type can be uncovered through single cell analysis. You also have the potential to uncover rare cell. For example, in the sample type, there could be some rare cell population existed, but this actually been masked through a bulk approach. So single cell sequencing actually has the ability to reduce rare cell to do rare cell discovery. And you also have the potential to do tissue atlasing. So if you're into high throughput, looking at tissue organ-wide kind of profiling, so our solution is very well adapted for tissue atlasing. And single cell analysis can also go into more advanced analysis, right? So you can do gene regulatory networks or lineage tracing, right? All this can be done with single cell analysis. So at Genomics, we actually have three foundational chemistry that I will introduce here. So the very first one you see here on the left is our universal assays. So your universal assays is the traditional or conventional 3' or 5' assay. So these are entirely poly-A capture chemistry. It requires a reverse transcription to transcribe out the full-length cDNA. And because of this workflow, right, our universal assays is compatible with long-read sequencing. So even to isoform detection, SNP detection, these assays can be quite useful for you to consider. So our universal assays also have a multi-omics solution. So with our 5' assays, right, you can actually do TCR, BCR profiling. So DDJ sequencing of these receptors can be done with our 5' immune profiling solution. You can do cell surface protein profiling, and even CRISPR screening. So I will not talk too deep into the universal assays, but I just want to highlight some of the standard application that we actually have came up in the past year or so. So one of the very interesting and exciting update for our Genomics universal assay is that we actually now allows you to do fixation. So in the past, our 3' or 5' assay is not compatible with fixation. And with this current demonstrated protocol, right, we actually allows you to fix your cells and also allows you to bring it downstream to do your 3' or 5' gene expression assay. So this demonstrated protocol has a very simple workflow. It's easy and also scalable with consistent across users. So we have validated these across disease and healthy samples. So not just PDMC, we have tested this on cell lines and other cell type as well. So these are quite compatible with a lot of different sample types. And because of these fixation capabilities, right, you can now really store your samples after you fix them at the -80 for up to a month before you can actually recover them and do your single cell analysis. So sample storage have a lot of advantage. First of all, you can now batch your sample, and batching your sample actually reduces batch effects if you are into when you do sequencing. So you can actually remove these batch effects because everything has been done in a single run. You can also save on cost, so you can actually multiplex or you can run everything in a single run. So this potentially can save you on cost for doing single cell. And speaking about multiplexing, 10x has also released a kit. This is what we call the on-chip multiplexing workflow, or OCM in short. So with this OCM kit, right, you're actually now supplied with 4 different color-coded gel beads. And this basically allows you to label individual samples. So with OCM, right, you can now multiplex up to 4 different samples and then collect them in a single well for the downstream library preparation. So after you sequence them, you can demultiplex based on the unique barcode being found on the cells. So this on-chip multiplexing workflow is very, very different from the conventional, site-seq where you actually label your cells with antibody, because this basically eliminates out any sample tagging. So you are actually saving on cost because you don't need to get the antibodies to label your cells, right, across the different sample types. And secondly, this is also compatible with cells that's not suitable for labeling, right? And then it's also flexible across many different sample types. And more importantly, it supports lower cell input compared to traditional multiplexing methods. So we can profile as little as down to 500 cells. And if you're doing multiplexing of 4 samples, you can actually go up to 5,000 cells per sample. So it's very useful for the precious samples like organoids or even limited cell numbers coming from biopsy, for example. So yeah, so on-chip multiplexing is available now. So if you want to know more about these kit, right, please feel free to reach out to us. All right, so second chemistry that I will discuss here is our flex assay. So our flex assay is our, it's actually very different from universal assay because our flex assay uses a probe-based chemistry. So remember, universal assay is based on poly-A capture chemistry, but flex, we designed probes to target the 18,000 protein coding genes in the transcriptome of human or mouse or rats. And because of this probe-based nature, right, it is very suitable for challenging samples like FFP or fixed tissues or fixed cells. So flex assay has also multi-omics options, so not just gene expression. You can also do protein and high throughput CRISPR screening, right, with our solution. So all this information coming from the same cell is possible with our flex assay. So last year in 2025, late last year, right? So we actually launched the latest and greatest Flex Apex. So this is our 3rd iteration of our Flex assay. So with Flex Apex now, you can do even more cells compared to our previous version. So in one single lane, if you look at this chromium chip here, there are actually 8 different lanes. In one lens, you can now profile up to 1 million cells per channel. So if you work out the math across 8 lanes, 8 million cells per chip is theoretically possible with our Flex Apex. And with Flex Apex, you also have this incredible multiplexing workflows. You can pull up to 384 samples into one single channel, right, and run this in a single run with our Chromium solution. So multiplexing really reduces the cost per sample, and you also can reduce the time and resources to do all these 384 samples across many different time points. So now you can all pull them together into one single lane and run in a single shot. And Flex Apex is also very, very modular. So our kits come in different, a kit pack has a lot of different components. And because of these, right, you can actually order whatever you have exhausted during your workflow. For example, if you run out of any of the reagents, right, you can still come back to reorder the particular reagents. And then from there, you can just complete your single cell workflow without reordering the entire kit. So it also offers flexible batching. So our kits, right, for example, the barcodes, right, you can actually freeze them after you use them for your single cell experiments. For example, this week you do 5 different samples, right? And then the next week you do 20 samples, and then the following week you do another 20 samples. So you can freeze and thaw these barcoding plates anytime you want. So we have tested this at freeze thaw cycle of 1 to 10 times. There's no compromise on the data quality. So yeah, so you can just run partial plates today and then save the rest for the later experiment you have. So it's very, very flexible for doing single cell without Flex Apex. So the other thing I want to share here is that in modern biology, right, one of the greatest visions in modern biology is this thing called virtual cell. So you might heard about virtual cell quite a lot of time. So a virtual cell is actually an AI model that can stimulate how a real cell behaves. And in order to achieve this, right, you need very large scale, high quality multimodal data sets. And this includes single cell transcriptomics data sets, epigenetics, multiomics, single cell perturbation, even spatial genomics might be actually required to do this AI modeling. So by integrating all these different modalities, right, the AI can actually start to learn the rules of cellular behavior. So ultimately, this enables in silico predictions, such as how the cell can respond to drugs, to mutation, or even environmental changes. And we actually talk about this since 2023. In 2023, when people talk about this, it still sounds like a fantasy, right? Because everyone wants to do this because you can actually do in silico prediction of how a cell behaves if you do a mutation in certain gene. But now it's getting into reality now, right? Because with our Flex Apex, you can do so much cells at such a lower cost, at such a very short turnaround time. It is now possible to do this AI modeling for virtual cell. So this is some of the commentary that's been published and review articles that have been published, right? So since 2023, there was a very first article, and this has been becoming a reality now in our modern science. So our Flex Apex is also transforming perturbation studies. So if you see here a preprint paper, you see on the left. So this is what a paper that developed this CRISPR screening called Proper-seq. And this is actually a single cell method of choice for training in the virtual cell competition by the ARC Institute. And then another very recent paper called the BI Perturb-seq, where this group of authors, they actually have a method to detect more than 57,000 perturbations with a set of less than 500 custom Flex probes. So what this means is that our Flex assay actually allows you to do custom probe design to do CRISPR screening, right? So with this particular method, right, you can use less than 500 custom probe to detect more than 57,000 perturbations. So this is an incredible method that they actually produce that can do high throughput screening and genome-wide CRISPR screening. And they also include intracellular protein co-detection as well. So yeah, so if you want to know more about this, feel free to reach out to this paper to learn more about that method. So lastly, the other chemistry in our Chromium platform here is our epi assays. So our epi assays basically stand for epigenetics assay. And with epi assays, we actually have single cell ATAC-seq, so you can look at open chromatin to investigate the epigenetic changes in your cell at a single cell resolution. We also have a multi-omic solution where you can combine both open chromatin and gene expression coming from the same nucleus and profile them, right? So this is our multi-omic capabilities for our epi assay. So we have launched the Genomics technology for epi multiome. So if you see here, some of the figures you see here in the middle. So the gene expression quality or the gene expression sensitivity has improved significantly with our Genomics technology compared to the previous version. And with the ATAC performance, right, we do see a very, very modest improvement. But nonetheless, you are still getting the highest data quality for a multi-omic solution, looking at ATAC and also the gene expression. So not just in terms of the improved data quality, right, we also improve on the scale. So previously you could do 10,000 nuclei, but with the Genomics technology now you can do up to 20,000 nuclei in per sample. And we also have reduced the cost for Genomics compared to the previous next-gen multi-omic solution. So, all right, I'm going to switch gear now to talk about spatial. So I'll start off with our Vision Spatial. So as we all know that a bulk approach or bulk RNA sequencing analysis approach, right, has this problem because you actually measure mixtures which can potentially be missed, which can potentially miss the underlying biology. Whereas if you do single cell analysis, right, you have the potential to list out every single component in your sample. So in this particular analogy, right, in these crushed metal pieces you see on the left, single cell analysis allows you to know exactly what components are being presented in this crushed metal. So we can see that this is actually a car, but of course, looking at what parts is available is important, but looking at where the parts are located is also very, very important. So this where spatial analysis is coming in. You can actually understand how the different parts or components are fit together to give you the full picture of your sample. So TEDx has come a long way for spatial biology. The very first Vision Spatial was launched in 2019. So we have been in around for spatial research since 6-7 years ago. So when we first launched Vision Spatial, our resolution is actually in the spot format. Every spot is 55 micron in diameters, and then there are gaps in between. So if you look at this particular example, so this is a FFP human colorectal cancer tissue sample on our first generation Vision Spatial data sets. And you take the same block and uses on our latest Vision, which is our Vision HD, which launched in 2024. So you can really appreciate the very, very high resolution coming from Vision HD. So Vision HD is no longer in spots format. So it's a 2x2 micron grid with no gaps in between. So every single, as long as your tissues is being captured by our capture area, so all your transcriptome can be captured without losing any information between the grids. So if we zoom into one particular region over here, you can also appreciate that our vision can potentially go down to the single cell scale resolution. So with HD, you can now resolve fine anatomical structure and cell type composition. So here is an example of a breast cancer FFP tissue. So if you see carefully, right, there are a lot of regions being circled in yellow. So these circled in yellow are actually the annotated tumor region or the DCIS region. So these are independently annotated by a pathologist. If the pathologists do not do any additional staining, like RNA staining for certain markers or cancer markers, right, you cannot tell what markers that these cells are expressed through an H&E image alone. But with Vision HD, you're interrogating the whole transcriptome. Any markers of interest that you're looking for can be reflected in your Vision HD data set. So if you look at these three images here, you can uncover what granular markers are expressed, where these markers are expressed. For example, the breast cancer markers, the HER2, ESR, PGR markers, you can easily resolve or see where exactly where these breast cancer markers are expressed in your tissue. And if you're looking into the DCI duct, the breast duct, right, myoepithelial cells, where you can see where the ducts are presented. So based on the marker these cells express, you can work out where these breast ducts are located in your sample itself. So VMHD is even more powerful if you combine single cell analysis. So this is just an example. If you can combine both single cell and spatial analysis together, right? So on the left is just a Vision HD unsupervised clustering. And if you combine single cell analysis, right, you can do things like tumor deconvolution, looking at tumor score, for example. So this is an example of a colorectal cancer tissue. You can see where the tumor cells are located. And you can even measure the tumor score based on the single cell data set projected into this Vision HD data set. So combining both single cell and spatial is not new. There's been a lot of papers that do this kind of deconvolution analysis. I just want to highlight this particular paper. So this is a paper that was published very, very recently. So this was in Nature Genetics in January 2025, where this group of authors, they performed single cell and spatial analysis to unlock key information on the oncology drug development program. So this is on lung cancer. So if you look at the cartoon on the right here, so this is basically a high-level overview of this Nature Genetics paper. They actually look into 19 different patients. So 6 of them are responder and 13 of them are non-responder. So they took tissues from these patients, performed single cell and spatial analysis, and they found that the responders, how the responder, the biology of the responders works. It actually has, the immune cell has the ability to proliferate and actually infiltrate into tumor region and to kill them. But more importantly, in the non-responder, right, they actually uncovered that the tumor cells together with the STP1 positive macrophage, they interacted with the cancer-associated fibroblasts to stimulate the deposition of collagen fibers at the tumor boundary. And because of these, right, they actually speculate that they obstruct the T cell infiltration. So the T cells can't penetrate into the tumor cell, therefore these patients don't respond to treatment. So it's only through single cell and spatial biology that you can uncover these very, very important insights. Then these can actually pave further ways to do drug development, how we can actually produce better treatment to prevent this kind of collagen fiber deposition in the tumor region cell. So another paper that is of very high significance. So this is actually in the AI modeling world. So this was published in Nature by some of our customers in Singapore. So if you know Joe Yong, so a very big spatial guy in Singapore. So they actually created this spatial immune scoring to predict the liver cancer recurrence. So what these clinicians and researchers did was that they actually took about 60 patients to develop a model development training to train this spatial immune scoring system. And then they actually apply this algorithm to another independent cohort for validation. So they look at more than 230 patients. And what they found here was that using spatial analysis and together with using vision spatial and other spatial technologies, right, they found that this AI modeling can actually better predict the recurrence than the standard clinical parameters. So they found that the NK cell enrichment at the tumor invasive fronts is actually associated with lower risk of recurrence. So yeah, so if you are interested to know more about this, please feel free to reach out to this paper. And in fact, this paper actually has a cover page in the Nature journal over here. So it's a very impressive article out there. So our Vision Spatial is compatible with a lot of different sample types. So disease samples, healthy tissues is all compatible. Even the way you prepare samples is compatible with FFP, fresh frozen, and fixed frozen. So our Vision HD is also compatible with mouse tissue or rat tissue, even non-human, non-human, non-mouse, and non-rat tissue. So if you have plant tissues or other model organism, right, you can actually potentially use Vision HD to do spatial analysis. So Vision HD has a lot of different capture areas. So when we first launched Vision HD, it was 6.5 by 6.5 millimeter capture area. We actually launched 11 by 11 millimeter capture area already. So if you want to have a better and bigger capture area, so you have the 11 millimeters for you to consider. So if you are working on tumor microarrays in a 1 millimeter biopsy core, a small capture area can perform up to 12 different samples. But if you do a 11 by 11, you can also do this, but you can do more than like 30 samples in a single capture area. So we also released a new protocol for FFP samples. So this has greatly improved the FFP sensitivity. So this is comparing the protocol 2.0 and protocol 1.0. With protocol 2.0, you can potentially uncover more transcript and more genes with this. So in the roadmap for Vision HD, very soon you will be able to profile proteins together with gene expression and morphology of H&E stain from the same tissue section. So this is currently in development. So very soon proteins will be enabled for Vision HD. So next, I will talk about our Xenium in situ technology. So as you all know here, Vision HD is entirely sequencing based. So you still need to incorporate sequencing as part of your workflow, and Vision HD interrogates the whole transcriptome. So whatever genes expressed, this will be captured, and this gives you an unbiased discovery method to doing spatial analysis. So the resolution for Vision HD is actually down to the single cell scale resolution. But for Xenium, in order to use Xenium in situ, you really need to know exactly what genes or what cell types that you're looking for, right? And Xenium is different from Vision HD because Xenium is imaging based. It doesn't require any sequencing. So the end-to-end workflow is actually provided from 10x Genomics, and also provide a very, very high resolution down to the subcellular resolution for Xenium in situ. Xenium has a very pretty big capture area. So if you look at the Xenium slide you see here, the measurement for the capture area is about 1 by 2 cm. So roughly this is how big a tissue you can place. And if you have very small tissue, like for example, a brain tissue of a mouse, potentially you can put up to 6 different tissues across 2 slides and run them. Or if you have a smaller tissue, you can potentially put more tissues within the capture area. So the idea is to put as many tissues as possible to make it a very cost-effective way to run Xenium in situ. Like for example, in tumor microarrays, so 1 millimeter cost, you can potentially run almost 200 different samples in a single Xenium run. So Xenium is compatible with a large variety of fresh frozen FFP tissue. There's no need to do any tissue optimization, right? So our protocol is universal. So most tissue types will be compatible with our Xenium workflow. So to prove this, we actually took 7 different cancer FFP tissues from the human brain, lung, skin, colon, pancreas, liver, and kidney. So we re-embedded them and sectioned them onto one single Xenium slide. And we can see a very robust data quality coming from our Xenium in situ overflow. And because Xenium is a targeted platform, so remember, Xenium doesn't do whole transcriptome. 10x actually developed pre-designed panels for some of the very common or popular tissues, for example, like the multi-tissue, the immuno-oncology panel, the brain, the breast, the lung, colon, and skin panels for human. We also designed 2 different panels for the mouse. And these panels range between 250 to 380 genes. And on top of this, 10x has also developed a 5,000 gene panel, or sometimes we call it the Xenium Prime 5K panel. This is giving our customer a more exploratory approach to look into your spatial data in your tissue. And on top of these pre-designed panels, right, we actually allow our users to add up to 100 custom targets. And what this means is that, for example, in the Prime 5K panel, there are 40 genes that's not included in the gene list. You can actually add up to 100 custom targets to spike into your probe, and then you can use this to run your spatial analysis. And we also fully support a full custom panel. So you can do up to 480 gene panel across any species of interest. So Xenium is species agnostic. So if you want to look on non-human, non-mouse, this is actually applicable for our Xenium in situ platform. And we also offer advanced custom. So for example, isoforms, xenographic exogenous sequences like viral transcript, bacterial transcript, SNP profiling, TCR, BCR profiling, these are all compatible with our Xenium in situ platform. So with regard to sample type, fresh frozen, FFP, fixed frozen are all compatible. We also have guidelines to deal with PBMC cell suspension, adherent cells, and even organoids as well. So here is just a snapshot of a Xenium data set. So this is what it looks like in front of a computer. So we actually have a software called a Xenium Explorer that you can download in your computer and explore our Xenium data sets. So this is an example of an immune cell interacting in the colorectal cancer of the tumor microenvironment on Xenium in situ. So you can see that Xenium can perform automated cell segmentation. So what we do here is that we look at the cell morphology, and then we do the cell boundary segmentation, and then assigning the transcript to within the cell. So if you look carefully, right, these different spots you see here, these are single molecule transcript detected by Xenium in situ. Every spot is one single molecule of transcript detected. And we know that this is a macrophage cell because it expresses these markers for macrophage cells. And we know that these are T cells because they express these markers, which is only found in T cells. So if you look at on the top here, this is a fibroblast cell. So fibroblasts are known to have irregular cell shape, right? We can actually capture this cell morphology very nicely and then segment the cell according to the cell morphology. And you can see this cell, this fibroblast is, in close proximity with the Treg cells on top. Just more data set to share. So here is looking at the entire transcript and the cell segmentation. So this, we can see that Xenium can actually perform very well with very dense transcript. So this is looking into the FFP human liver tissue on the left, and then the FFP human breast on the right. So Xenium allows you to do both RNA and proteins profiling, right? So if you are into proteins on top of gene expression, right, we actually have a cocktail of proteins that you can choose from. So this is this protein comprises of 27 different proteins. So it involves the immune cell typing A, B, and C, the proliferation and differentiation proteins, the immune checkpoint protein, and the tumor microenvironment proteins. So with these 27 proteins, you can mix and match. You don't have to do every single one. So if you want to do only immune cell typing A, you can just use this protein to do the Xenium analysis. So I'm going to share with you here. It's an example of a proteogenomics specific insights coming from Xenium in situ. So this is a renal cancer tissue, a grade 2 renal cancer tissue. a post-genome H&E image. So you actually do H&E imaging after you run your genome on the same tissue section. And the one is you see circled in black are the tumor invasive region. So this is actually identified by pathologists at where the tumor regions are located. So if you perform in situ analysis for transcript, right, here we can see that of these tumor regions, right, there's actually one tumor region that has a transcriptome profile significantly different from the other tumor regions over here. And then if you apply proteomics readout, right, so using proteins, we actually saw one particular tumor region over here having a protein profile different from the other tumor regions. So this is how powerful you can get if you combine both gene expression and proteins from the same tissue section to uncover more insights into your tissue. So next, finally, I will going to talk about our latest and greatest from 10x. So this is Artera. So Artera is another in situ platform. So you can actually visualize Artera as the bigger brother of Xenium in situ. So whatever Xenium can do, Artera can do as well, but Artera can do more. So with Artera, we will now allow our users to do whole transcriptome profiling in situ. So imaging-based whole transcriptome profiling. We are also going to allow a much higher throughput compared to Xenium. So you can do up to 800 whole transcriptome samples over 1 cm square in a single year. And we also have improved on the sensitivity and specificity of this in situ platform, all at the subcellular resolution. So with Artera, you now no longer require a specialized chip or specialized slides from us. So you can use off the slide from the lab. So any most standard microscopic slides will be compatible with Artera. We allow for scalable batches, meaning that you can batch and process small samples with one single prep, so they can maximize your instrument walk-away time. It has a bigger capture area. So if you remember, Xenium is about 2 cm square, but for Artera, you're looking at more than 5 cm square capture area. And we also have a stackable panel menu for this platform. So Artera, when we are shipping this instrument this quarter, right, we will allow our users to do whole transcriptome, so 18,000 protein coding genes in human. We also allow our users to stack up to 1,000 gene custom, and mouse whole transcriptome will come next. And if you don't want to do whole transcriptome, right, we also allow our users to do an Artera select. So this is basically our pre-designed panels. So this is stackable 1,000 gene panels. We can stack up to 3 different panels for up to 3,000 gene targets. So human multi-tissue will be available at launch, and soon there will be other panels available. And we also allow our customers to do full custom. So you can do up to 3,000 gene targets, and this can be any sequence in any species. So here is a preview of our Artera whole transcriptome assay. So in this particular slide you see here, there is about 6 or 7 different tissues that are cell type we actually tested on. So if you look at this cell palette, in single cell level, right, we can actually detect across more than 10,000 transcripts per cell. And these are actually very dependent on your tissue and cell type. So you can do, you can really regularly see transcript exceeding more than 1,000 in a per cell resolution. So Artera deliver single cell sensitivity. So if you see this graph here, the y-axis on the left is the Artera whole transcriptome assay, and on the x-axis is the single cell flex apex data set. So we tested on the FFP human testis. So we take this tissue block, section a few curves to do flex single cell gene expression, and then one code to do the whole transcriptome. you can see the correlation to single cell analysis is very, very high. So we're looking at odds of about 0.75. And in fact, if you look at it carefully, right, Atera seems to be performing a little bit better from the single cell assay. And what we want to drive this message here is that the sensitivity of Atera is comparable to our Chromium Flex Apex assay. So here is just another data set from Atera. So here is we are looking at a human brain FFP sample, and the one in yellow is the 18S interior stain. So we stain the interior of the cell using an 18S fluorescent probe, and then we also stain the DAPI to do the cell segmentation. And in this particular sample, we can really capture a lot of transcript from the individual cell. And some of these cells actually exceed 30,000 transcript. Right, so for example, in this cell, we're looking at 39,000 transcript in one single cell here. So you can work out what markers are expressed. You can use this marker to identify what cell types these are. So for example, the inhibitory neurons over here, the excitatory neurons over here as well. All right, I will just spend the next 2 minutes to talk about how you can use Atera to do tumor microenvironment analysis. So here is an example of a cervical cancer tissue FFP. This is actually a HPV 16 positive tissue. And over here, we are showing the morphology staining. So the DAPI stains blue for the nucleus. We also have the 18S interior stain to stain the interior of the cell. And then the pink is actually the boundary stain, and the green will be the interior stain for the interior proteins. So basically, this morphology stain is to do cell segmentation. And in this particular data set, we detected more than 700,000 cells. And within these 700,000 cells, there is actually more than 1 billion transcripts decoded over here. So basically, we profile here, there's actually more than 20 different cell types that we detected. And if you look at this UMAP plot on the bottom left-hand corner, you can see this very beautiful clustering of different cell types are uncovered with the Atera. So Atera can potentially power biomarker discovery. So what we found here was that there are 2 different tumor types, subtypes in this tissue. So one of them is the hypoxia tumor, and the other one is the proliferative tumor subtype. So we actually found that there are certain markers only specific to hypoxia. So for example, ODEM is something that we found that's only specific to hypoxia tumor, and the FGFR3 is maps to the proliferative tumor cells. And more interestingly is that these different tumor subtypes are mutually exclusive from one another. So they are found in one region and not the other. Right, so what's next for Atera after we launch this in this quarter? So there's a very exciting roadmap for Atera. So whole transcriptome analysis will be extended to not just human, it will be to mouse and other species as well. The software will be upgraded. There will be the workflow automation, so end-to-end workflow automation for slide preparations. I'm very excited about the targeted base-by-base sequencing. So you can now eventually do in situ sequencing of a short region to uncover mutations or other variants that they're interested in. So and protein profiling will definitely come along the way. So here just a snapshot of what the in situ sequencing can be like. So you can uncover a specific mutation like IDH2, C to T mutations. All these can be done with eventually with Atera. And proteins, spatial protein characterization will be also made available very soon. So here is just a snapshot of proteins staining on top of gene expression. And eventually, we also have an end-to-end workflow automation. So this will really reduce the hands-on time and also the total slide preparation turnaround time. So yeah, so this is something that's going to be very exciting for a lot of our users. So we believe Artera will be the key to resolve biological complexity. And this is because we can provide the resolution and scale to make deeper insights with full spatial context from molecule to tissue. All right, so I'll end this here. So I'll be happy to take any questions from the floor, and I will thank you for your attention.
Yeah, thank you, Benson, for the insightful presentation on how single-cell and spatial multiomics are transforming the research applications, along with the latest innovations from 10x Genomics. Now we will move into our Q&A session. You can unmute yourself to ask a question or feel free to type in the chat box. And Benson, we have a question in the chat box from Ethan. How stable is the fixed cell? How does the fixed assay works? Isn't fixation lead to fragmented RNA?
Sorry, I'm looking for the chat box. Where is it?
Yeah, it should be in the right, bottom right.
Ah, okay, I saw it.
Yeah.
Ah, okay. So yeah, so fixation has been a concern for researchers in the past, right? So if you want to do single cell, usually they don't fix their cells for doing single cell. But now with our flex assay, you actually fix yourself with formaldehyde, and formaldehyde are known to be quite harsh to the RNA molecules. Basically, they fragment them. But because our flex assay is probe-based, the footprint is actually only 50 base pairs. So although RNA can be fragmented at multiple different locations, right? But as long as 50 base pair remains intact, that the probes can bind, so then this will be detected by our flex assay solution. So we actually have tested FFP tissue blocks at more than 10 years old, and there's no problems with our flex single cell assay. So our flex assay can deal with very, very challenging tissues like fixed tissue or FFP tissues.
Okay, we have another question from Louis Xiang. How does your Chromium platform compare to instrument-free workflow? What are the advantages?
Yeah, so there are a lot of, the few instrument-free single-cell platforms out there, but we believe a platform is still advantageous to those instrument-free platforms because, first of all, it reduces. Okay, so first of all, it do the partitioning at a very fast format. So you do your single-cell partitioning in just 6 minutes. If you do an instrument-free workflow, right, there's a lot of pipetting. There's actually rooms for error. And what I know is that in the event that one of the wells are not being filled up properly, right, then you basically waste the entire kit. So there's a lot of inconsistency and reproducibility issues with instrument-free platform. So with a platform, you can be assured for reproducibility and consistent in your workflow. And more importantly, the data quality with the platform is actually much better than instrument-free platform. So these are some of the main advantages for Chromium compared to instrument-free platform.
Okay, thank you, Benson.
Yeah.
Another question from Elaine is, what kind of support does 10x provide after the post-sales?
Yeah, so this is very important, right? So we're not selling products and then we don't take care of you after that. So post-sale support is very, very important for us. In fact, we actually provide a very, very comprehensive support post-sale. So we actually have different teams out there. We have a tech support team. We actually have an application support scientist. We even have a bioinformatics team to deal with all the different questions or help that you need from us. We also have an engineer team. So if you have a platform, we have a POS service engineer to come to you to fix your instrument if there's any problem. So comprehensive post-sale support is provided from 10x. So if you have any question like troubleshooting devices, feel free to write out to us, 10x support at 10xgenomics.com. We will be able to reply and help you with your problems.
Okay, thank you. Another question from Yi Fui. How does Artera and Xenium convert images to transcriptomics information? Is it solely based on staining from samples?
Yeah, so this is actually not an amateurish question. It's actually a very good technical question. So I didn't delve into the technical part of it. But basically what happens in Altera or Xenium, right, is our chemistry here is that we use a single-stranded DNA probe that binds to your transcript of interest, the targeted transcript, right? And then there's a rolling circle amplification to amplify the capture sequence. So fluorescent probes will come in with the amplified product, and this image, right, will be captured by our system. So if you think about fluorescent in situ hybridization, where you label your specific targeted transcript with fluorescent probe, right? Xenium and Altera is very, very similar. It's just that these two systems, right, they can look at thousands of genes, like for Altera is 18,000 genes at one time. Yeah, so it's imaging based. It's not sequencing based. I hope I answered your question. So it's not based on staining, if that's the question you're asking.